Advanced Fit technology utilizes different textures in the silicone skirt to create a superior seal against the diver's face. This unique feature, found exclusively in aqualung masks, also minimizes the marks left on the skin after a dive.
Best Dive has more than 20 years of experience in developing and manufacturing wetsuits. We use 100% Yamamoto neoprene with exclusive material license in China. Freediving champion Jessie Liu is our global ambassador. We collaborate closely with international and domestic freediving events. In 2018, Best Dive was awarded the title of the most popular domestic brand by ADEX China. We offer a variety of styles and high-quality services. You are welcomed at Best Dive.
High Springs, Florida. Population, 5,941 people. Located just south of the Santa Fe River. Home to springs, caves, and divers. Home to housing. We are small-town America. We have big hearts, and we take pride in the work of our hands. Here at Halcyon, we understand everyone has their own reason for diving, and we understand each one of those divers is looking for the equipment that best suits their dive. We understand every detail, every stitch, every nut and bolt and screw that we put into our gear. We understand you desire the best and are not willing to settle for anything less. That's why every product at Halcyon is tested and approved for both our dive and yours. We take things personally, fine-tuning every piece and packaging each product carefully by hand. Our 8 to 5 is for your weekend dive. We are here day in and day out so you can experience the underwater world. It's simple. This is why we do what we do at Halcyon. Because, just like you, we understand underwater.
you. Hello, everybody. Thank you for taking time to this webinar, Passion Make Possible Uniquely Singapore, and it's the Ocean Science edition. So, thank you, moderator and all the panelists for taking time to share with all the audience and us about this very unique Ocean Science edition. So, I would like to first start off to introduce myself. I'm Carter. I am the organizing team for ADEX Pixel Virtual Expo. And because due to COVID-19, ADEX cannot do the show physically. Therefore, we have converted this ADEX to APE, which is ADEX Pixel Virtual Expo, and connect the diving community and underwater community together while we tide over this pandemic. So now, I would like to introduce our moderator tonight. Her name is Shu Qin from Singapore. Hello.
Hi.
Sam. So she is a research assistant at Tropical Marine Science Institute. She is also a co-founder of our Singapore Reef. All right, Sam, over to you, please.
Okay, thank you, Carter, and the ADEX team for the opportunity to moderate this session. It's really very exciting because we are going local. I think for the first time, we're actually showcasing marine science in this platform. So today we have with us these 4 amazing ladies who have dedicated a lot of their time in marine research and conservation, not only locally and also regionally. And I really look up to each and every one of them very much. Okay, so before we start, I just want to ask, how is everyone doing? How is it like to be stuck on land during this period?
Dry.
Yeah, we're really dry.
Writing papers?
The Prof. Charles set you up for that.
I also turned on the arrows. I'm writing. Okay, so it's going to be quite exciting for this session. We are going to hear more about their research and work on the environment and organisms within, particularly seagrasses, corals, fishes, and turtles. Okay, and to those who have been saying there's nothing to look forward to in Singapore, this session is going to blow your mind. Okay, so without further ado, let me introduce our panelists. So first up, we have Dr. Yan Xiang. Okay, so Dr. Au is a research fellow with the St. John's Island National Marine Laboratory. So from 2012 to 2015, she went diving at Great Barrier Reef and completed her PhD education along the way. I love this. Okay, while studying physiological responses of tropical seagrasses to declining water quality and ocean acidification. So now her current research interest lies in understanding how marine organisms such as corals and seagrasses interact with their changing environments. Okay, so next up we have Dr. Jani Tanzil.
Hi.
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 So, Dr. Jani Tanzil is a marine ecologist with Tropical Marine Science Institute, National University of Singapore. She is also one of the resident scientists working at St. John's Island National Marine Laboratory. So her current research involves examining the bands of the skeleton of coral. So something similar like looking at tree rings to reconstruct past environment conditions, looking at the history, and also to determine the growth rates of corals around Southeast Asia. So with her work, Dr. Tanzil hopes to better understand coral responses to climate change and other environmental disturbances. Okay, so next up, we have Dr. Zeehan Jaafar.
Hello.
Hi. So Dr. Jaafar is a marine biologist at National University of Singapore. So her research interests are in the evolution and ecology of marine fishes and also about the conservation of their habitats. So Dr. Jaafar has led and participated in many international and regional expeditions to enhance understanding on the diversity and distribution patterns of fish fauna. She is also the lead editor of the Singapore Blue Plan 2018, which her team worked with the marine community to put together a set of recommendations to preserve Singapore's marine landscape. All right, so Dr. Jaafar also enjoys sharing her passion for the region marine area through public engagement and also education. And finally, lastly, we have Dr. Karenne Tun.
Hello.
Okay, so Dr. Tun is the Director of Coastal and Marine Branch at the National Biodiversity Centre, National Park Boards. So Dr. Tun is also actively involved in coordinating coral reef monitoring efforts in the Southeast Asian region. So Dr. Tun's current work at NPAC covers issues related to management and conservation of Singapore's coastal and marine environment, and also the biodiversity that it supports. So her team is also responsible for managing Singapore's first marine park, which is the Sisters Islands Marine Park, and it works closely with the wider community to encourage responsible stewardship and sustainable use of the marine park to achieve the overarching objective of conservation, outreach, education, and research. Okay, so now let's get going with the sharing from our panelists. So first up, let's have Dr. Yan Xiang to share about seagrasses. Dr. Ao, please.
Okay, thank you, Sam, for the very generous introduction. Okay, hang on, let me share. Okay, not wait. Wait. Sorry. Share screen. This one.
So while Dr. Ao is setting up, I think you can, if you have any questions, please direct the questions to on the Facebook page.
Okay, I'm set up finally.
Okay, let's go.
Okay, hi everyone. My name is Yan Xiang. You can call me YX. I'm a marine scientist who looks at the biology and ecology of seagrass ecosystems. So, compared to seagrass beds, everyone knows a lot more about coral reefs, how beautiful, how colorful, and how lively coral reefs are. So my one of the first few impressions I got from Great Barrier Reef was that how magnificent the marine life is. But when we zoom out and look at the continent of Australia on its own, we realize something fascinating about the marine environment in the northeast Queensland coastline, which is the Great Barrier Reef actually forms a barrier that really effectively blocks the wind and waves coming from the Southern Pacific Ocean and the Coral Sea, thereby creating a very relatively quiet lagoonal area behind it. So this creates a very conducive environment for seagrass beds to form and for seagrasses to thrive. And it was there at the Great Barrier Reef that I've ironically fell in love with seagrasses and decided to just go ahead and do seagrass research. So, so this is me doing most of my seagrasses. So most of my PhD work revolves around running mesocosm experiments. So what mesocosm means is that I bring the organisms out of their environment and put them into carefully controlled tanks, where I can manipulate the seawater conditions to mimic various stresses such as climate change. So some of my PhD focuses on the responses of tropical seagrasses to ocean acidification. So ocean acidification is caused by increasing levels of CO2 dissolving into seawater. And what that does to seawater is, as the name suggests, it acidifies it. And it creates not so pleasant environment for calcifying organisms in particular. But my research actually shows that ocean acidification does benefit seagrasses in terms of increasing its photosynthetic rates and also growth rates. But this is also contingent on what the prevailing water quality is like, how much light and how much nutrients these plants have access to. So coming back to Singapore, I wonder how can I parlay the findings from my PhD results into something more usable and application specifically. So that's how I came up with this project to look at how seagrasses can help coral reefs or adjacent coastal ecosystems that are under threat from climate change. So the basic principle of this is that since seagrasses are going to do well, are projected to do well under climate change, they should be able to take up the extra carbon dioxide from seawater through photosynthesis and in a sense push pH towards the more alkaline range. And in doing so, a more alkaline environment will be more conducive for corals to grow and thrive. So, so the work that I've embarked for this project is quite similar to what I did for my PhD, which is also running mesocosm experiments. But the main difference this time is I was able to secure some funding to hire younger and more able bodies to do the work. Yeah. So coming back to the title of this project, of this slide, as a scientist, I'm very aware of the privilege I have being able to work intimately with the organisms and falling in love and understanding the beauty and appeal of grass, not very appealing, but yeah. So sometimes I wonder to myself, like, how can I actually translate all that beauty for laymen, people who do not have access to all that scientific information as readily as I do? So I want to do it through pictures then, maybe. So we have to remember that seagrasses are important. Actually, they are the only food sources for some very charismatic large marine organisms like dugongs and green turtles. So in Singapore, we do have a small green turtle population, and we also see dugong trails in our seagrass beds from time to time. So this shows that seagrass beds is actually probably sustaining a population of these large charismatic marine animals. And besides, this seagrass beds actually host a lot of small, tiny critters. Sometimes they are very hard to see, like your mollusks, like your crabs, like your sea urchins. And all these tiny little organisms form very important chains in the complex food webs in seagrass beds. So sometimes if you are diving or walking along a seagrass bed, you might be lucky enough to chance upon the apex predators, such as a stingray, or even a wobbegong shark. That one was very cute. He was just lazing there, chilling out, and just letting us take photos of him or her. I don't know. Okay, and besides, seagrass beds are also home to many commercially important marine species. So the coral reef species that we eat, such as snappers and mullets, the baby and teenager fishes of these species, they nurse and they feed in seagrass beds before they grow big enough and mature enough to swim out to the coral reefs. So finally, I want to end my slides with this photo. What I want to show is that basically seagrass beds are happy places for many marine creatures. And I hope that it can be a happy place for you too. So it is my tiny little wish that more people will just go out there, capture and showcase the beauty of seagrass beds in the region, so that there's more incentive for people to be more aware and more willing to conserve these very vulnerable natural habitats. So thank you.
Thank you, Dr. Au. That was so nice. Make me feel like I really want to go back to the field.
Me
Okay,
too.
yeah. Okay, so some question from me. So for, let's say, people who are actually interested in finding out more about seagrasses, can they actually go to these fields? Do you need to dive to get near to the seagrasses?
So if we are talking about Singapore, in the context of Singapore, most of our seagrass beds, they tend to be quite turbid when there's water over them. So how we scientists typically assess them is to apply for a permit to get to the field site intertidally. So when the tide is out, when the land is dry, we'll just walk out and do our research and surveys. But in terms of diving or snorkeling right now, I think the water is still a bit turbid. Like, not so easy, not so welcoming for beginners, I would say. Yeah.
Is there any questions, Carter?
No, let's move on. We'll curate the question later.
Okay. So for now, shall we move on to Dr. Yani? So she'll share her work on coral. Dr. Yani, please.
Okay, so I share screen.
Mm-hmm.
Okay, hold on. Okay, can you see?
Yes.
Okay, so hi, I'm Yani. I'm a marine ecologist, and I really work a lot with corals. So actually, some people ask me like, why do you only want to work on corals? Why do you like corals so much? Well, I like it because, other than the fact that it's pretty, it's colorful, they're also key ecosystem engineers of the coral reef ecosystem. And I like to think of them as the underwater engineers that build self-repairing, ever-increasing HDB flats that marine life can call home. So they're very important, and that's why I particularly think they're important to study as well. So some of the corals, my favorite coral to study, and what I really my research is focused on, are these large, long-lived corals. This particular one is called Big Mama in the American Samoa. It's 6 meters high, 41 meters in circumference, and thought to be over 500 years old. So some of these corals can grow very big and live for a very long time. So what do I do when I see them underwater? Some of these corals, if you cut them in half, right, these large corals, what you see are bands in the coral skeletons that are akin to tree rings. And using these bands, what we can do is date them and then go back in time to reconstruct a variety of things, including their past growth rates. So here, using the bands, we can look at their growth patterns and how their growth rates have changed over time. And we did this for a bunch of corals around Southeast Asia, particularly Malaysia, Thailand, and Singapore. And what we had found was that since 1980s, the corals around this region, the Paraites corals specifically, have declined in growth rates by about 20% over the last 3 to 4 decades, which is a little bit worrying because the rate of growth of corals actually relate to how fast the reef can recover as well. That the HDB blocks that the marine life called home, the structural framework of it all. And this decline in growth rate we found was also related to when you start seeing sea warming rates increasing as well. So the decline in growth rates are what we think related to climate change and especially sea warming. So other things that we can use the coral skeletons for is that when the corals grow, they incorporate things into their coral skeleton, like trace elements, strontium, calcium, barium, and all sorts of other things. So we can use the geochemistry in the coral skeleton to reconstruct the past environment in which they live. For instance, if you look at the colorful plot here, you see that the lead to calcium ratio over time, that was actually measured in the coral skeleton, and it was used to reconstruct the past lead concentration or lead amounts in the environment over time. I particularly like this graph because you can kind of see that the lead calcium ratio, which reflects lead in the environment, increased over time. And then in the 1980s, 1990s, you see there's this decline, which coincided with which basically was when they decided to ban lead in leaded petrol, and we see a decline in the lead in the coral, which is reflective of lead in the environment. So it kind of shows like what we can do as a community, as a society, if we reduce certain pollutant in the environment through proper management, we can also possibly reverse the effects of climate change and other sort of not very good emissions of things. The other plot that you see is actually a plot of the strontium calcium ratio in the coral that plotted against sea surface temperature. You can kind of not be able to tell the two different lines at all, because the strontium-calcium ratio in the coral is a very, very robust proxy of sea surface temperature. And this is what paleoclimate scientists have actually used to reconstruct past temperature using corals and past temperature in the marine environment using corals. And of course, we can use a whole bunch of other geochemical proxies in the coral skeleton to reconstruct past environment, assuming we have the proper in situ measurements as well to calibrate these proxies. So a lot of the times I'm asked, like, how do you core corals? How do you actually get these corals to go to analyze their skeleton? So I thought I might just share with you a little bit how we do this. It literally is taking a drill underwater and working with a good team. It's a team effort to core the corals, to obtain a nice core from these large, long-lived corals. We don't bring the whole giant colony up. I think that's impossible, and it's also very destructive. So the first thing we do usually is we get the team to sort of set up a drill stand. So you can see how large these corals are, these long-lived corals are. And then once the drill stand is set up, we proceed to core using a hydraulic corer underwater. And again, I think there's a lot of people here because usually when this happens, when somebody takes a video of something, it is when we have less heavy work going on. So we core the corals using these hydraulic drills, and we core it in sections, 30 to 50 centimeter sections, and then we break it. We use a core catcher to extract the section, and then we sort of extend the coring barrel, and then we keep coring deeper to get very long cores down to the base of these large, sort of 4 to 6 meter tall coral colonies. So it's very heavy, these extensions, because seawater is very corrosive. We use steel so that it doesn't rust, but they're very, very heavy. So there needs to be a lot of manpower to work together underwater when we're coring. So this is actually what the coral sections look like when we bring them up. Okay, we check if the growth axis is good, it's nice, the connections are good as well. And then if it's all good, and then we're happy with whatever we get, and then we continue on. And then once it's over, we've reached the base, and then we wrap everything up, and then we surface. So that's not the end. After we get the coral, we don't just, that's it, that's goodbye to the coral. We do a little bit of TLC to the coral. We try to leave no trace, clean up after ourselves underwater. Here you see people on the boat. It's a very, very big team effort for this. And then what we do is we also plug the coral holes, the coral core holes, with a cement plug or marine epoxy, so that the corals can recover. And they do recover very quickly. And then within a year or so, you don't actually see that. Don't see any evidence of that the coral has been sampled or cored. So these are like some of the coral cores that we get from the coring efforts. For some reason, people like to measure themselves against the long cores. So here we have like a 200-year coral core obtained from Sarawak, around 2.3 meters. And then this is the longest core that we've ever sampled together with a good friend of mine from Malaysia, Jani, and from Pulau Tenggol, 4.2 meter cores. And what we do is we slap them, we process them, and then we date them. So you can see here, we look at the bands, and then we date the coral to go back in time. And reconstruct like past growth rates and past environment. So with the coral core, with these long-lived corals and these coral cores, what we can do is go back in time. We don't need fancy machines to go back in time. I don't think we have any of them anyway. But we can use the corals themselves and the skeletons of the corals specifically as time machines to look at how they have fared in the changing environment, and also how the environment has changed over time. Yeah. Okay, so let me just stop sharing. Yes.
Thank you, Dr.
Okay.
Yani.
Yeah, thank you, Dr. Yani. I believe we have some questions from the audience. However, I believe there are quite a few presentation and interesting presentation to move on. So we will, as the panelists go on and present their research, we will take all those questions at the end of this panel discussion. So Sam, let's move on.
Okay. So actually, can I ask Yani one question?
Sure.
Okay. I want to know, like, what is the oldest coral that Yani has ever found?
Or the oldest one that I've ever found?
Yeah.
In Singapore or whatever?
In Singapore first.
Oh, in Singapore, I think the oldest one I've ever caught a coral is about, and it goes back to about like 1950s, 1960. And the oldest one that the 4.2 meter coral core from Tenggo was the oldest core we've gotten. I think it goes all the way back to the 1700s. So that's what, 300 plus years?
Wow,
yeah.
That's crazy.
400 years, yeah.
Okay, thank you. That's very interesting. So now can we have?
How about Sam? Just one question for Dr. Au. It's from Teo. Teo, are there efforts to enhance the conditions along our beaches to encourage the growth of these seagrasses given its ecological importance? Dr. Au, please.
Okay, I think that's a very interesting question. Firstly, I don't think our shorelines have been deliberately manufactured or manicured to encourage seagrasses to recruit. But miraculously, we do have seagrasses recruiting to our coastlines naturally, because essentially they are plants. They just need enough light, suitable sediment, something muddy, something sandy. It's like a good mix between them to grow. So a lot of our coastline is actually very favorable for seagrass to grow. So as long as these places are not disturbed too much, seagrasses will be able to establish if they are able to get there to the site. Yeah.
Okay. Thank you, Dr. Au. So now let's continue with the presentations, and we have Dr. Zeehan Jaafar to tell us all about fishes. Dr. Jaafar, please.
Hello, hi. Thank you, Sam. Thank you, everyone. I would like to take this opportunity to thank the edX team for having me here today. It's always a pleasure to share our work with everyone. So I am going to start sharing my screen. What I'll do is I'll talk a bit about a project that a group of us have been working on for the past 2 years. It is called Codefish SG, and this project is carried out with funds from the National Research Foundation, and we are carrying out this project together with team members from Temasek Life Science Laboratory, headed there by Dr. Henning Seedorf, also with our counterparts at Lee Kong Chian Natural History Museum, and our collaborators at the National Parks Board. So what we are trying to do here is we are trying to create, or we are creating, a library of fishes that are found in Singapore, and this library is of their DNA. Now, in Singapore, we have more than 800 species of marine fishes. So our marine fishes are actually quite well known, but not all of them are substantiated, which means that some of these names or some of these species have been reported since the 1800s, but we have not seen them since. So not all 800 species are valid. Today, we know more or less about 500 species. What we have done for this past 2 years, and will continue for another year, is that we are taking DNA samples from these fishes, and we are sequencing them. And these sequences from these DNA samples, right, we upload to a database. What happens then is that this database, as you get more and more species coming in, will act like a reference library. Now, over the years, we've been increasing. So now we have almost 500 species that we have barcoded. Okay, so we use two markers. We use 12S as well as the CO1. These are, markers that are used globally to identify organisms. And we have taken these sequences and uploaded them to a database. Now, we collect our fishes through various means. We, use nets, as you see here. We use, we dive. We use scuba gear as well. And we also collect baby fishes. We do this all over Singapore. And we continue to do this. It's actually quite a difficult thing trying to get fishes and trying to get their DNA, but we try our best in not hurting the fishes. Usually when we're carrying out research, there's a lot of permitting processes that we go through. And there are also a lot of ethical things that we have to think about. So whenever we work with organisms, we have to ensure that we comply with global standards in ensuring that the animals do not suffer, right? So, so now we have a bit over 500 species that we have barcoded, and I think we're in good shape, and we are curating this. Now, what will then this fish library be used for? So it has a really broad use, right? So other than just looking at the DNA, we also take photographs of the fishes. We make sure that the fishes are identifiable so that other people can use this data, right? The library itself, the sequences, once they are up in the library, in the future, if we have organisms, for example, fish larvae, which means our baby fishes or juvenile fishes, and we don't know what they are because juvenile fishes look very, very different from the adult fishes. So these are some photographs here of juvenile fishes, right? So baby fishes and young fishes, it's very, very different from the adults. So very difficult to identify to species. But if we have the species of the adult in the library and we take the DNA of the juveniles or the babies, then we can match them up and we can know what they are. And in the future, we can then identify them. Also, a lot of fishes carry out phase changes. They may change sex, may change color and appearance throughout their lifetime. So this DNA, but their DNA will stay the same, right? So this DNA library will also then help us identify them. Sometimes there are also species that look very similar, but they are actually different species. This is what we call cryptic species. And using their DNA, we are able to tell that they are two different species rather than one. In the future, with a very good database, a very good library system, we can just take water, okay, and run the sequences which are found, all that DNA in the water, because fishes would shed scales, they would shed slime into the water, and we take all that DNA and we can sequence it, and without catching the fishes themselves, we can actually know what are the fishes that are present in the water. And we've actually done some test runs around Singapore, within the mangroves, within the reefs and seagrasses, and we've had very good information coming out of it. Through this method, we can also then look at invasive species. We can know whether there are invasive species in our waters without actually, looking at them, without actually seeing them, right? Through this method of the eDNA. Also, with this kind of library, if, for example, you have a fish fillet in front of you, but you're not sure, the guy who's selling you the fish fillet says, well, this is actually a really expensive fish. You want to know what it is, you can take a bit of the DNA and you can, if the library is very good, you can compare it to the library and you can match the name and you can know what that fish fillet is, right? So there are many, many uses of the library. These are just some of them. In the future, once this project wraps up, we are actually going to make this DNA library open to the public. Okay, so I'm going to just end off this presentation. It's going to be a short one, and I welcome any questions later. But I would like to end this presentation by posing a question to you. How can you help the fish library? And I think we are always very, very appreciative and very grateful for any help that we can get. You are really our eyes on the reefs as well as the waters. Please contribute any photographs, any specimens, especially if you're angling. any rare records that you might find, or important behaviors. So photographs can tell 1,000 words. This is, for example, a photo from Miss Heng Pei Yan, and it shows a cardinal fish. This is a male cardinal fish who is mouth brooding. So all those tiny eyes that you see inside the mouth of this male fish are little baby fishes. So these kinds of behaviors, it tells, one picture tells 1,000 words, right? So it's amazing, and it's really helpful for us. So you can send your queries and your information and your photographs, this email address. This is my personal email address, and I will answer each and every one of you, each and every one of the emails. So we would be very, very grateful for any information that you wish to share with us. So thank you very much.
Thank you, Dr. Zeehan Jaafar. And also, please take note of the opportunity that you can actually get all your pictures, especially during this period, curate all your pictures of fish, and then you can send to Dr. Zeehan. Okay, so, and then you can, in a way, you can contribute to science as well. So we also have a question from the audience. So they're asking, are these fishes only found in Singapore?
So currently, because this is a grant meant for Singapore, we are actually focusing on Singapore fishes, but we hope to expand this to the regional fishes. Especially because Singapore is such a big seafood consumer, right? A lot of our fishes come from the region and from abroad, right? Globally. So, having a fish database that can support the kind of fishes that we get in Singapore, especially through the food industry, is actually very important. But this kind of effort will, really corroborate a lot of the efforts that are being done elsewhere. So if we can get more of that regional and global information, of course, that will be the best. For now, this particular project in Singapore, but we're hoping to expand in the near future. Thank you.
Okay, so I think one more question.
Sure.
Okay, sure. Just one more, then we move on to Karen. Yeah.
Okay, so we have one interesting question. Are bull sharks native to Singapore? Or are they seasonal visitors like the birds in Sungai Buloh?
It's very difficult because, as you know, waters in Singapore are very turbid. It's quite difficult to track these kinds of apex predators without satellite or tracking devices, right? But we know that they're native. We have had many records of them historically, but we also continue to get records of them at least once or twice a year. We know, for example, there was someone who had angled the bull shark of Changi last year and then earlier this year of Labrador. So they are there. They are in our waters. But so far, the ones that we have in our waters, they've not been very big. Yeah, they're still juvenile.
Good to hear. Okay, thank you again. So last but not least, let's move on to Dr. Karenne Tun. So she's going to share about her team's efforts in marine science and conservation. Dr. Tun, please.
Okay, hi everyone. I just unmuted myself. Let me share my screen. Okay. Can you see the screen?
Yep, clear, very clear.
Okay, great. So I would like to start by thanking ADEX team for organizing this. I think it's very meaningful. Also the opportunity for us to share with the general public what's going on, especially in these times when very little fieldwork is going on. People might forget that there's actually a lot of work going on. So it gives us the opportunity, and I really thank the team for that. So what I'm going to share today is a snapshot of what we're doing at NParks under the Coastal and Marine team, and I put it as working with the community to safeguard Singapore's coastal and marine environment. I'm a biologist, marine biologist by training, but over the years, the area that I really enjoyed most was not so much the primary science, the fundamental science, but the applied science, the applied research. I worked at NUS, similar to all the other panelists who are here. I also work within the region, coordinating coral reef monitoring. And one of the things I found is there's a lot of good science that's going on. But then for science to become effective, it needs to be somehow translated to management, where managers, policymakers, people without the scientific background can use it. policy should and needs to be guided by good science, and I think that is an opportunity that not many people get the chance to do so. In 2013, when the opening came out with NParks, for me it was a no-brainer. It was an opportunity for me to finally realize that transition from science to management. And over the years, I have an amazing team of folks to work with, a very small team of 12, and this team, we work together to basically safeguard the entire coastline of Singapore. Singapore's coastal and marine environment, working with our collaborators, fall under a very small team of 12, very passionate team, and because of them, I think we could have achieved a lot in the last decade or so. But the question that we always get from a lot of people is, what are we trying to conserve? What do we have left in Singapore? Most people don't even realize coastal and marine environment. So the question is, what are we doing? Why are we doing it? So I would like to start this presentation by just quoting an inspiration. My professor, Professor Chow, also the professor for every single panelist here. He said something many years back that will always be the mantra that keeps me going. And he says, in relation to coral reefs, but I think it's applicable across, all habitats. He says, if our reefs haven't given up, then why should we give up on them? And I think that's really important for all of us doing the work, because sometimes we might feel very challenged in the environment that we're in, but if those fellas out there that we are working with haven't given up, then there's no reason for us to. So with that, I'm going to start. Basically, how do we manage our coastal and marine environment in Singapore? We have something called a Nature Conservation Master Plan. This is an actual concrete plan that basically articulates how Singapore's vision in conserving nature in Singapore, and this includes terrestrial and marine. Let me see, is it moving? Yeah. So basically, the way it is structured, it has 4 thrusts. The first thrust is conservation of key habitats for the marine environment. And now we have the Sisters Island Marine Park, but then we also have Labrador and Sungai Buloh up in the north. In Thrust 2, we look at habitat enhancement, restoration, and species recovery. The mantra that most scientists will preach is, habitats have an ability to recover. As far as possible, we don't touch them. We try to protect them as it is and let them function. But in an environment like Singapore, where you have very limited habitats, sometimes areas need a bit of help. And that's why we come in through habitat enhancement, restoration programs, and certain species that might need a little bit of boost to help them recover. We have a species recovery program. Now, all this is basically underpinned by very strong research, applied research in our context, because the good science is needed, and the good science is then used to apply it into management. So applied research for conservation, biology, and planning is one of the keystones of the work we do. And finally, and to me, it's really one of the most important, is community stewardship. Bringing it all together, we need the community to be involved. And we're not only talking about the academic community, who are the pillars, but the general public, people who are out there who interact with the environment. If we can reach out to them and get them to love the environment, marine environment, terrestrial, both, as much as we do, then we have a chance to really do something to protect it. So that's basically what the Nature Conservation Master Plan is. It is the master plan for Singapore. And under this, we have what we call the Marine Conservation Action Plan. So taking a broad master plan, and we drill down to actual action on the ground for the marine environment. We have the Marine Conservation Action Plan. It's quite a comprehensive plan, and we don't have time to talk about it now. So I'll just talk about one aspect of it, which I think is really important, and it goes back to my background, which I started off with, which is monitoring. So monitoring is basically one of the activities that underpins the 4 thrusts in the Nature Conservation Master Plan, the Marine Conservation Action Plan, which is to document changing states of our habitats and biodiversity over space and time, to identify priority conservation areas and species, to also identify nuisance and threats, adjust our existing management plans, or come up with new plans and conservation strategies as and when needed. And as we apply them, to evaluate its efficacy of the strategies that we have, and also to assess compliance with international conservation agreement. So this is basically what we do in a gist within the Coastal and Marine team. Again, small team of 12, we can't do all of this, especially the research. So a big part of our work is working in collaboration with the broader academic community. I'm glad to say that all the panelists here are our collaborators, and we are working on projects with them. So I'm really proud to be, connected with them, as well as so many others across various institutions. So they form the basis for the science that goes into the policy and planning. So what I'll do in the next 2 slides is just talk about some of the work that we are doing at NParks itself. Mostly monitoring the really nitty-gritty science is done by the science. Monitoring. So one of the core work we do is coral reef monitoring, and a colleague of mine, Jeffrey, runs a citizen science program called Reef Friends. He goes out several times a year to monitor our coral reef. So we go there to look at reef community. We also have a program on the Neptune Stock Sponge, and one member of the team, Jani, she's doing a master's that's actually studying the growth rates of the Neptune Stock Sponge. Besides the routine monitoring, we also do ad hoc seasonal situational monitoring, like coral spawning. In this little video that I showed earlier is the bleaching monitoring. So if we have any kind of indication that the bleaching might occur, and sad to say that the indicators are this year, our reefs might be at stress, and we are really hoping to get out as soon as we can to go and monitor the reefs in case there's a bleaching event. So we document the bleaching, we document recovery, important aspect. And just as we recovery from bleaching, we also document coral spawning, which is the other aspect. If corals can spawn, if we can produce the next generation, then we know that our reefs have a chance. So every year, with the exception of this year, because circuit breaker measures didn't allow us to go out, we've been going out to monitor coral spawning and our reefs. And this was taken, I believe, last year during the 2019 spawning. The other aspect that we're very proud of, we have a really dedicated team, Karenne Tun, Jod Eng, Yan Xiang, Jonathan Tan, plus the rest of them who are involved in the Marine Turtle Conservation Program. It's a program that really only started 3 years ago, and the team has really achieved amazing results in these past few years. They went overseas, they went to Mont Repos Turtle Hatchery in Australia to learn everything from A to Z from turtle hatchery management from the experts there, and they've come back and they've applied it here. So we have a turtle hatchery at Sisters Island, and whenever a nest is observed by a member of public or during some of our beach patrol programs, they'll go out and monitor. If the nest needs to be moved, they will move it. They will monitor the temperature, and when the hatchlings emerge, they will ensure that the hatchlings have imprint, get out to sea. Every year, if we can get a few hundred hatchlings out, there's a chance at least one of them will return in 15, 20 years' time to continue the next generation. So in the last 3 years, I think an amazing amount of data has been collected by the team with the support of the Marine Turtle Working Group, which is a group made up of so many individuals from the community that contributes both time, effort, as well as knowledge to the program that we have. Aside from that, we also try to encourage citizen science programs within the community. Cho Pei Wong runs the Intertidal Watch program with many of the colleagues and interns who have come and over the years. There's a long list, and I can't remember all the names, but this is really a labor of love that was started several years ago. And there's an incredible amount of data that's been collected. Over 400 volunteers that have been trained. And these are just your general members of public who have been trained to do see the intertidal watch across 4 areas. I think, if I'm not wrong, maybe even 5 areas in Singapore now. And this was quite important because when there was the oil spill event in 2019 to 2018, they went out to Changi to monitor in December. Oil spill occurred in January, and they went back again in February to monitor, and they found that thankfully there wasn't that much change. So monitoring like this, even though it is done at the citizen level, is so important to generate the kind of data to show that if we can go out and monitor, then we can assess whether there's really an impact from events that happened, and if there was an impact, what can we do to intervene? And lastly, I'll end with another baby of ours, the Friends of the Marine Parks community. To me, this is one of the most important communities because it actually brings together all stakeholders involved in the marine, from academics to NGOs to interest groups. And our purpose really is to come together and work as a community to address issues related to the coastal and marine environment, whether it is biodiversity impacts, surveys, whether it is oil spill. The community comes together, we find solutions together. If there are conflicts with usage of the environment, for example, recreational use versus conservation, rather than have a headbutt session, come together, talk about how can we achieve a common goal together, where every stakeholder has a role to play, also as a part of the environment that they can use and enjoy. So the Friends of the Marine Park community is really something that's growing, and I think in the next few years, you will see some exciting programs that will come up with it. Again, I'm happy to say that many of the panelists are part of the Friends of the Marine Park, running many of the programs. So I'm really grateful to the commitment, contribution, and dedication of this community. And I think that's my last slide, and I'll end here. Thanks.
Thank you, Dr. Tun. That was so informative. And it's really heartening to really hear that, you guys are including science-based approach and public engagement in coastal management. So we have one question from Parizat. So she understands that the largest artificial reef structure was built on Sisters Island around 2 years ago. Is there any news on the progress of it? And do you see this as a viable solution to restore our coral reefs?
And thank you very much for the question. The Artificial Reef Project, quite an exciting project that was done in collaboration with JTC as part of their corporate effort to give back to the environment that we use quite a lot. So the whole idea was to create structures that can mimic the natural environment within the water column. So there are shallow areas. Again, it's a man-made structure. It took a while for it to stabilize. So within the first year since it was established, there was a transition of animals and plants that grew on it. So you had algae, you had barnacle growing on it. And in the last 6 months or so, very happy to say that we started seeing coral recruits that are growing. Some of them are almost as big as a palm size, so it's good to see that. Again, it took about 6 months for it to stabilize. So there wasn't that much settlement from the spawning that occurred last year. So hopefully after this year's spawning, we'll see a greater variety of species. It's also a huge structure that is a great aggregator of fish. And I think Dr. Zeehan has a research project on it, looking at how it is contributing to the fish community. Again, I would like to highlight the Marine Park is a park that is there for research, conservation, education, and outreach. And one of the core ambits of that is a no-take area. We want to make sure that the marine park can serve a purpose to enhance biodiversity. And fish is one of those really charismatic group that if they have a place that they can call home, they will reproduce there, but their offspring can almost never stay in the same environment, and they spread out. So if you can protect even a small area, and we're talking about a very, very small area in the Sisters Island Marine Park. Now, if that area can serve as a great nursery ground for fish, then it will actually contribute to all the other areas. Fishermen don't need to harvest from this area. They will have continuous harvest from St. John's, Lazarus, and all the other areas. So that's one of the is to serve as an additional home, basically a nice safe haven for the fish community. So it's serving multiple purposes, still very early on to say how it is progressing. If you look at a lot of literature, you need 5-10 years of data before you can even assess the success. So we are still there, again, working with the research community to find some of these answers to see whether it's really effective. And then if it is, then the next stage, we might then look at designs, optimize the design that are a bit better reflective of the environment, perhaps. We look with the scientific community for that, for the next phase. Thanks for the question.
Thank you, Dr. Tun.
Dr. Tun, I think from the audience, Sean Goh has one more question. It says, hi, Dr. Karenne. Are there any plans to incorporate nature-based solutions into the Marine Conservation Action Plan?
Great question, and definitely a big yes to that. So again, I didn't show you the Marine Conservation Action Plan. It's really quite a huge, complex plan that includes all aspects of it. Nature-based solutions is really coming up as one of the key approach moving forward as we tackle climate change, sea level rise, rather than just focus on engineered structures, which would be required in some instances, but then how do we then apply nature-based solutions where we can? So again, priority would be nature-based solutions. If not, then how can we enhance built areas with nature-based enhancements to it. So we are looking into that. I think there are several groups that are looking into that. NUS has a new unit set up, a Center for Nature-Based Solutions. If I'm wrong, Dr. Koh Yan Pin has just come back to Singapore. This is one of those guys who spearheaded some of these. So yes, the short answer is yes, and we are working with the broader scientific community to achieve that. Thank you.
Okay, thank you, Dr. Tun. Okay, so here's a question for Dr. Zeehan. Okay, so I have observed large congregations of bait fish like glassfish and anchovies around the pillars of Bedok Jetty. So will they be more dispersed in the sea around underwater structures, such as man-made structures, or they will be congregated near shore?
You have to unmute.
Thank you for your question. With glass fishes, anchovies, hardyheads, they occur in typically in schools. They are attracted to structures, but you can also find them near shore, especially at high tide. They do congregate in the shallower waters. So they do occur in schools in the shallower waters as well. But you tend to see them around the jetties more because these large structures are aggregating devices for these fishes, which also underpins the importance of big structures like the artificial reef, which Dr. Karenne Tun was talking about at Sisters Island Marine Park. You also see large fishes aggregating at these man-made structures as well.
Okay, thank you.
Now we have one more question for Dr. Zeehan. From the Marie Tan ask for the eDNA detection, possible even with transient species in Singapore waters.
Okay, so I think Marie Tan, you're asking if using this eDNA technique, whether we can capture the presence of transient species, right? So species which are not there all the time, but are moving around. It really depends on how much that organism sheds, how long their DNA stays in the system, whether there's very high currents through the system, through this area, or if that amount of DNA just sits down and settles, if that area is, there's hardly any movement of water. So there are many, many factors that can affect the result of this. Sometimes when we also have large storms and fresh water comes from upriver, push down river, and it congregates at the river mouth and around the river mouth, you also can pick up DNA from fishes upriver. So sometimes you get fishes which are from the fresh waters. Sometimes if someone eats a salmon by the beach and then throw it into the water, you get salmon DNA. Sometimes if someone eats chicken wings by the beach and then throw it into the water, you get chicken DNA, right? So it really is a whole bunch of things. And then you have to, again, look at the library, compare what is there and see whether, this is salmon actually really found here? Could it be that it's a food fish brought in? But so there's many, many different factors that affect that, right? So for example, we also do eDNA we look for dugongs through eDNA. Okay, so a student of mine is working on this project, and you can actually find dugong DNA in the soil samples that we find around seagrass areas. So Dr. Yan Xiang and I are working on that together with the student. So, and dugongs are transient, they move around a lot in order to forage. So yes, in a way, you do get these transient species, but it is dependent on the factors that I mentioned just now. Thank you.
Thank you.
Dr. Zeehan, that leads to one more question personally from myself. Have you worked with any forensic scientists if there is kind of corpse or deaths? It's a leading from your answer, the question to answer, and then you mentioned that if fish eats certain kind of things at that point of time, within a certain period of window, we can sequence that. Am I correct to say that?
Yeah. So, but that goes the same for someone who swims, right, in the water, and if they shed skin or hair, you can pick that up as well. Because what happens is what happens with eDNA is that you take a volume of water, so typically 3 to 5 liters of water, and then you spin it down so that you basically filter everything, and you just take the DNA material. So all that water is filtered out, and then you sequence whatever material that is there, and then you match it with with the DNA libraries to find out what they are. So anything that's been in the water and whose DNA can survive that area, that period or that time, then they appear in your database or in your data.
Interesting. I think Karen has a question for Dr. Au. Yeah.
Yes, I do. So, Dr. Au, I have to say that seagrass areas are really understudied and appreciated compared to coral reefs and mangroves, which have a lot of attention. And yes, seagrass really play an important role. They are the middleman right between mangroves and coral reefs. The other important aspect of seagrass is the blue carbon aspect of it. So we understand a lot about the blue carbon of mangroves. Perhaps in your opinion, what do you think are the potential for seagrass to have to contribute to blue carbon in Singapore?
Okay, so in Singapore, the studies on blue carbon in Singapore seagrass meadows, it's still its nascent stages. We have several groups in National University of Singapore who are looking at the carbon stocks of seagrass beds, and I do think that at least our seagrass beds have a potential contribution to our carbon. Carbon stock, the carbon stock of Singapore, in terms of accounting for how much carbon we can prevent from emitting to the atmosphere. So more studies need to be done on that, but there is a great potential because we are a highly biodiverse environment, and our seagrasses are quite robust and resilient to stresses.
Okay, thank you. Okay, so we have one last question, and this is open to everyone. So how can the public, or how can we actually get involved in such work, and what will be the relevant qualifications required in Singapore? Anyone want to take this question?
Maybe I can start, and then others can join in. Again, like I mentioned, at least for NParks, and I speak for NParks, we do have several citizen science programs. So if you go to the NParks website, it will link you to some of the programs, whether it is the Turtle Beach, the Turtle program, we call it the Biodiversity Beach Patrol. We've also got the Intertidal Watch, but not just within my team, but across NParks. There are quite a few citizen science programs that the general public can get involved in. So please go to the website, check it out, and sign up for it. We have over 40,000 volunteers, believe it or not, across NParks. Many of them are terrestrial gardeners and stuff, but it's a growing community, and it's a community we really want to encourage and nurture and grow with together. Thank you.
So I can add into that with the so as part of this project, trying to build this DNA library for Singapore marine fishes, we had an expedition in Singapore in August last year, and for that expedition we actually included more than 100 volunteers over 12 days, and we invited many people, many stakeholders Part of them are actually from the Friends of the Marine Park group, but there are also volunteers who, for example, who like angling, and they continue to volunteer for us even though we're out of the expedition phase. So there are avenues, and if you're interested to help, please do email. It is not going to be possible during the circuit breaker period or maybe even slightly after, but as things improve, we would be happy to have you help us in our research. We actually seriously really welcome any help that we can get. For me personally, outside of the project of these marine fishes, fish photographs, especially from waters in Singapore, are actually very valuable for us. It allows us to tell a story and to tell people what's there. And often, as you can tell when we're working, it's not really possible to take photographs and then let alone nice photographs, right? So having these will really, really help us. Yeah.
Anything from Yani and Dr. Au?
I think the same would be for corals as well. I think like having, like Zee says, having eyes on the ground would be really good. On the reef, that's right, on the reef, underwater, because we can't be everywhere at the same time. I mean, everywhere all the time. So, for instance, with coral bleaching, if you see anything, out of the ordinary, you see something weird, signs of coral disease, for instance, or even large corals that get you excited, like, it does me, then, you can just reach out to us and let us know, like, it enriches our scientific library as well from knowledge from people on the ground. Yeah.
Thank you.
So for me, I think what I really look for in volunteers is really the willingness to get down and dirty, because seagrass work is not exactly very clean, very glamorous. You will emerge looking like a beauty queen. And also a good attitude is also very important. A good attitude in asking and learning and approaching researchers in a respectful manner, and not demand for information. So yeah, I think we are all, like what my fellow panelists have mentioned, we are always looking for volunteers, looking for help, looking for eyes on the reef, on seagrass beds. So as long as you're willing to work hard and willing to work together in a team, I think that's enough to be involved in marine research work.
Can
Thank you, Dr. Au. Yes, please.
Yeah, I wanted to add, other than, working in the field, I think a lot of misconception is that, hey, you've got marine ecologists, you're out doing diving, doing field work, but actually a lot of our time, we spend data entry, doing really, really boring things like data entry, data analysis, staring at the computer, measuring surface area on image J or something. So, we need volunteers, willing volunteers to help do all these other things as well. So, if you have other skills that, that are not necessarily, diving related, you can't go out to the field, but still want to contribute, and you have computer skills and willing to sit down and, crunch the numbers for us, we're up for that as well.
Okay, so thank you. So like what our panelists have said, no efforts are too small, and really anyone can contribute to marine conservation. So do reach out to any one of us if you're interested in marine science or conservation, and we'll be happy to direct you to the right person. So once again, we thank you all, Dr. Yan Xiang and Dr. Jani Tanzil from St. John's Island National Marine Lab, Dr. Zeehan Jaafar from National University of Singapore, and Dr. Karenne Tun from National Park Board for your time and wonderful insights. And of course, thank you to all our friends who have stayed throughout the session with us. So I guess this is where we say goodbye, and we hope you learned a lot from this session. So we look forward to seeing you and all of you again. Thank you.
Thank you, Sam.
Thank you. But well, before we leave this live webinar, I have one last thing to share. Let me share my screen before we leave. But of course, I would like to thank moderator Sam and all the panelists, doctor panelists, for all the insightful and very, very informative sharing I think this will be a very good video for the public to read, watch, and learn more what of all the citizen science have done towards contributing to ocean. So last but not least, I'd like to share this slide. We have our World Ocean Day, World Ocean Week from Monday to Tuesday. On Monday, on the 8th of June 2020 is our World Ocean Day. On Tuesday will be our Coral Triangle Day, which is on the 9th of June. And Wednesday will be our Blue-Green 101 topics, which is on the 10th of June. And Ocean Odyssey 101 on 11th of June, Thursday. And Friday, Scuba 101, 12th of June. So stay tuned, and more exciting and interesting webinar will be coming up online. Last but not least, thank you again for everybody's present, and we shall meet each other online very soon.
Okay.
Bye.
Bye.
Bye.